Greetings and welcome to the United States Transhumanist Party Virtual Enlightenment Salon. My name is Gennady Stolyarov II, and I am the chairman of the U.S. Transhumanist Party. Here we hold conversations with some of the world's leading thinkers in longevity, science, technology, philosophy, and politics. Like the philosophers of the Age of Enlightenment, we aim to connect every field of human endeavor and arrive at new insights to achieve longer lives, greater rationality, and the progress of our civilization.
Thank you, Hannah, for that introduction. Greetings, ladies and gentlemen, and welcome to our final panel of RAADfest in 2022. Today, you will be hearing about some of the most ambitious technological endeavors that are poised to transform the human condition in the following years and decades. And the innovators you will be hearing from today are involved in an extremely diverse range of fields. However, I think there is one overarching concept that could help you in your minds unify these endeavors in terms of what they portend for the future of our species. And that is the idea of transhumanism. Now, transhumanism has many different interpretations, but I would say the most accurate and the broadest interpretation of the term transhumanism is the use of science and technology to fundamentally transform the human condition for the better, making us more human, better versions of ourselves without losing anything good about our humanity. And transhumanism can take the form of innovations in biology. It can also take the form of innovations in biotechnology, nanotechnology, robotics, artificial intelligence, a wide range of endeavors. And quite fittingly, our speakers today are men of numerous talents. They are polymaths, including our first speaker, Dr. Nuno Martins, who is a well-known polymath. He is a researcher, he is an entrepreneur, he is a public speaker, he is a life and health extension supporter, and as a polymath, he usually likes to make use of different subject areas, drawing ideas and concepts from different bodies of knowledge to solve specific problems. And you'll hear this today in his talk on brain-computer interfaces. Please welcome to the stage Dr. Nuno Martins.
Hey, thank you. Thank you. Thank you. Hey, everyone. Ready for the talk? Let's go for it. Ready, let's go. So, there we go. What future are we creating? What is the truth about a post-longevity world? And the main question I have for you is this: can we create a better world together? What you see here are the 17 Sustainable Development Goals. If we solve all of these goals, if we achieve these goals, if we solve these challenges, in principle, we will create a better world for everyone. No poverty, no hunger, water for everyone, good health, education, and so forth. Let's look into goal number 3. Good health and well-being. That's exactly why we're here. We're trying to create a world where everyone enjoys a healthy, long life. So we're working to achieve goal number 3 of the Sustainable Development Goals. Question: will human healthy longevity become a reality? It's scientifically possible. And we have several approaches to achieve that goal. Epigenetic rejuvenation, senolytics, genetic therapies, telomere-based approaches, thymus regeneration, microbiome optimization, parabiosis, just to name a few. Is money supporting these scientific efforts? There are billions invested in longevity today. There are hundreds of longevity companies solving the challenges. So either all of these brilliant scientists, investors, and entrepreneurs are completely wrong, or it will become a reality. Human, healthy longevity will become a reality soon. And we'll learn that in this amazing conference. Question: can we enjoy a long, healthy life in a world with poverty, hunger, and war? Can we really be happy? If not, can we achieve these 17 Sustainable Development Goals? Can we create a world without poverty, with water, food, a shelter, healthcare, and so forth, for everyone on the planet? Let's look into goal number 4. Quality education. What if we had a technology that would allow us to educate everyone around the world? If every single person in the world would be knowledgeable and would understand science, technology, it's very likely that locally they would be able to take care of their lives and the lives of those around them. That means that there would be no poverty, there would be food, they would be able to find a way to produce food locally, provide water to one another, create a shelter for one another, and create a local economy, as we have in the States. The question is, how can we solve education? Will we ever build a university in the poorest areas of the world? Universities help educate everyone, our population, schools and universities. But there are more than 1 billion people in the world living in the poorest areas of the planet. Will we ever build a university there? Likely not. So how can we educate? How can we democratize and universalize education? 3 years ago, we published a paper titled Human Brain Cloud Interface. How to connect the human brain with the cloud directly, without the use of a cell phone or a laptop. What was interesting was that the paper is now in the top 10 most viewed papers ever on a very prestigious journal called Frontiers of Neuroscience. And the journal has more than 10,000 published papers. But more than being proud of the success of this paper, what this means is that the scientific community recognizes that now is the time to create this technology. So let's do it. How can we do this? Let's look into technology and science. What do we know today? What's our body made of? Cells, right? We're all made of cells. Next question. What are cells made of? What you see here is a motor protein carrying a vesicle. This is happening right now inside many of your cells. The kinesin, this motor protein, is a biological nanorobot. and we have many of these inside our cells. And it's not just the motor protein. Our cells are built of small nanomachinery. This is the most accurate representation of a cell that we have today. Next question. Can we see and move and maneuver things at that small scale? 10 years ago. This was 10 years ago. IBM produced a movie. It's called A Boy and Its Atom. Every single dot you see on this image is an individual atom. So 10 years ago, IBM and other labs around the world were able to move atoms one by one. And an atom is way smaller, much smaller, than the motor protein you just saw and that moves inside your cells. That was 10 years ago. Well, what are we doing today? These are some nanocars. This is a competition held annually where scientists build these nanocars atom by atom, and they compete. They make a race to see who wins, who's faster. So essentially, scientists are playing video games. That's what we do with the government's money. Well, you could say, this is great, but these small machines that we're building today, can we interface them with cells? What you see here is called the sperm bot. It's picking up an individual sperm cell. It's moving the sperm cell along. It's giving it mobility again. And as you see here, it's helping it fertilize an egg. This is how human life starts. This is how your life started. So let's assume for a second. Let's assume that we would be able to build today a series of nanomachines, and we could use them to interface with our neurons and connect our brains to the cloud. Would we be able to extract the information from our brains into a supercomputer? Put it differently, what's the processing power of the human brain? 5.52, this is a paper we published 10 years ago. 5.52 plus or less 1.13 times 10 to the 16 bits per second. This is, okay, 1,000 is 3 zeros, 6 zeros is a million, 9 zeros is a billion, 12 a trillion. This is a quadrillion. This is 50 quadrillion bits per second. 10 to the 16 bits per second. That's a lot of data per second. Question, what's the fastest supercomputer today? 1.1 quintillion operations per second. That's 10 to the 18 bits per second. That's 20 times faster than a human brain today. I'm not saying this supercomputer is smarter than a human brain, but it could handle all the data coming from a human brain. So if we use this technology and we have a thought, we have a question, we could send information from our brain to the supercomputer and it would reply in real time, giving us an answer. This is today. Supercomputers are faster than the human brain. This is great, but this is going to be academia, right? This is all a bunch of scientists doing some stuff in the lab. Have we ever built a prototype? What you see here, this lady is feeding herself, controlling a robotic arm just by thinking. And this device, being built at UC Berkeley, not our lab, another lab at UC Berkeley, next to ours indeed. And this is a device that allows us to connect with our nerve fibers, and this is the data that we extract in real time from the nerve where it's connected. It's extremely biocompatible, can be implanted for decades, it's totally fine. And this demonstrates that as of today, we already have a prototype for one specific nerve cell. Next question. Will there ever be a company crazy enough to bring this to market? Let me introduce you to Mr. Elon Musk. He recently created a company called Neuralink. What's the vision of Neuralink? To connect the human brain with computers. Well, but that's Elon, right? Nobody else in the world is doing this, right? There are more than 188 neurotech companies literally around the world. So let's assume for a second that this will become a reality. We will connect the brain with the cloud in the near future. How will this change education? Do you really need to have people on a room and feed them with information on classrooms? Should we focus on memorizing data? Or should we focus on teaching values? Teaching how to pursue happiness. Teach how to love one another. But more than simply access the cloud, can we upgrade our intelligence? Can we become smarter? When we connect the brain to the cloud, we're connecting the brain also to the artificial algorithms that run on the cloud. Just recently, an artificial intelligence algorithm was better than a board of 6 certified physicians diagnosing cancer. Can we become smarter? And if we're smarter, then we can solve the challenges we have ahead. So the conclusion we had on the paper is, it's possible, it's desirable, and it's very likely to happen. We will build, in the next couple of years / decades, human-brain cloud interface systems. Coming back to the first slide. Can we create a better world together? That was the first question of this talk. I believe so. And this community is a great example of that. Together, we're building a better world. A world where we're all healthy, can enjoy life, and supported by these technologies, these brain-cloud interface technologies, we're very likely to live in a much better world. A world where we can all pursue happiness and enjoy life. My website, just in case you want to get in touch. And to finish, I want to thank you for giving me a little bit of your most precious asset, your time. Thank you. Thank you. Thank you.
And our next speaker is going to outline an innovative approach to do just that. He is Dr. Jean Hébert. He is the professor in the Department of Neuroscience and also the professor in the Department of Genetics at the Albert Einstein College of Medicine. His research is focused on devising methods of cell replacement for the adult neurocortex where its cells are lost due to damage from age-related degeneration. Ladies and gentlemen, please welcome Dr. Jean Hébert.
Thank you. Hello. First, I'd like to thank the organizers and James and Bill for inviting me. Thank you. I'm going to tell you a little bit about our work today, but I first want to put it into a context that I think is important for us to understand in terms of not just the aging brain, but aging in general. So I'll spend a little bit of time on that and then go over a little bit what we're doing. So why and then how we're doing what we're doing. The title of my talk, Progressive Brain Tissue Replacement as a Key Component to Beating Aging, sort of implies that there's a surgical approach here if we're replacing tissue. So that's kind of a scary thing. Why would we want to do surgery on our brains? And that's what the first part of my talk, I'm going to try and tell you why that's something that I think we'll have to confront at some point. All right. So aging, there's a lot of different definitions of it, but the one that I think is important to consider is sort of one of the core processes that are often overlooked when we are considering aging. We look a lot at performance, phenotypes, how well, how fast we can run, or how metabolically active our cells are, how well they're producing things. But those are sort of downstream effects of aging. At the core, what aging is an accumulation of damage. And I'll describe that in a little more detail here. So our bodies are made of not just cells, but also space between the cells. That's the extracellular matrix. And we know that damage accumulates in both, and probably more so in the extracellular space between the cells, that it's non-metabolic or has very low metabolism, and so there's not very much repair processes to maintain things in good shape. And a lot of the components of that are made very early in life and just sit there for our whole lives and accumulate damage. So those are, that space is filled by proteins and carbohydrates. But DNA also, damage also occurs within cells. So in DNA, for example, we know of mutations. It gets a lot of attention. Also occurs to lipids. So those are sort of the four components that make up our bodies: protein, carbohydrates, DNA and nucleic acids in general, and lipids. And they all accumulate damage over time. And the problem is that this damage is complex and stochastic. Right, so it doesn't occur at the same place in each protein, even if we're talking about the same protein, like a particular collagen, for example. It'll happen at different places in the collagen. And the variety of types of damage that occur to these proteins is quite complex. So here's an example just for proteins of these types of damage that have been shown to accumulate with age. And the damage to carbohydrates, which is another component of the extracellular matrix, hasn't even really been explored in any detail yet. So there's likely to be, carbohydrates are much more complicated than the proteins that exist in the extracellular matrix. There's 1,000 possible proteins that exist in the extracellular space. But carbohydrates, the number is much greater. They're much more variable. There are these disaccharide concatemers that can be very large, and they likely accumulate damage as well. And we haven't even begun to think of how we might address that damage. So the main point I want to make here is that addressing this damage is essential to beating aging. If we just keep ignoring this damage that accumulates, we might be able to enhance our performance as we age and compress morbidity, but we will not be able to beat aging. So you may ask, well, maybe instead of taking a surgical approach to replacing tissue, maybe we could just fix all that damage with drugs. And I think that's a little overly ambitious, again, because of the complexity of damage. Drugs are not that smart. They'll target one particular target, and because this damage occurs stochastically, that target will look different for each protein in each place. So you would need hundreds of different types of drugs that don't have side effects to be able to reverse this damage. So that's less likely to be a plausible approach to addressing this problem. We hear a lot about gene therapy. Okay, maybe we can just express genes that will target these forms of damage and replace them. But again, there exists, as of now, no machinery, no genes that encode proteins or the machinery necessary to repair this damage. Right? There are proteins that can turn over some components of the extracellular matrix when it's not damaged. For example, collagen, most abundant protein in the body, which is why I come back to it. It accumulates damage, and it is no longer recognized by any of the existing machinery to degrade it and then possibly replace it with new collagen, for example. So we would have to not only invent all this new machinery to recognize this damage, but also to turn it over and then somehow deliver that machinery to all the cells of our body, and particularly difficult for the brain. That's also something that's going to be challenging at best and difficult for me to imagine how that might work. Another approach is replacing cells. There are cell transplant treatments that people are developing. But again, we've done this experiment before where we take young cells. This is true for the brain. It's true for other parts of the body. We take young cells, we put them in an old body, and they behave like old cells. And it's not surprising because this extracellular matrix is very important to their well-being. And if it's degraded, the cells just don't perform like they're supposed to. And we've done the reverse experiment too, where we take old cells and put them in a young body. And those old cells will behave more like young cells. Again, confirming the importance of the niche surrounding these cells. But replacing the cells themselves is not going to be enough then. We need to also address the damage that occurs in cells and the damage that occurs around cells. And how do we do that? So, we go from the small drug molecule level, that's going to be very difficult, to the gene level, to the cell level. The level that will work is a tissue replacement approach. So that's what we're focusing on. So tissues and organs have been replaced throughout the whole human body, usually to treat diseases or accidents, trauma, damage due to trauma. But basically every part of the human body except the brain has been surgically replaced, either with biological replacements from deceased donors, which is not a source that we're proposing here, but to save a life, this is what happens. Sometimes it's a synthetic replacement, so we know of artificial hearts and artificial limbs now that are outperforming natural limbs. Sometimes it's a hybrid between a bioreactor and a synthetic machine. There's half artificial, half biological kidneys that are approaching clinical trials. But there's a whole field of regenerative medicine that's working on body part replacements. And surgically, it's possible to implement that. And although it's been used so far just to treat diseases and repair trauma, if you do it at a more wholesale level, you will rejuvenate the body or get rid of all that old damage. The problem, of course, is the brain. So you can't replace the brain. It's who you are, at least not as a whole organ. So I'll talk a little bit more about that. And every part of the brain is important and will need to be replaced. We're focusing first on one part of the brain, which is the bigger part of the brain. This part that's labeled with the little asterisks, that's the neocortex. So that's the part that encodes our highest cognitive functions, much of our self-identity. It's what we want to preserve, possibly more than any other part of the body. So I'll focus on that. And so does it even make sense to think of progressively replacing neocortical tissue over time to remove the old tissue and put in new young tissue? And the answer is yes, and that's based on two very well-established findings in neuroscience. One is a very old finding that the neocortex is extremely plastic in the sense that functions can move from one location to another seamlessly without us knowing. And so I'll give you an example of that I think is pretty striking, but there's quite a few examples of this, and it's been shown across mammalian species, including humans, and it's been shown for all the different functional modalities that are encoded in our neocortex. The second fundamental reason why it's possible to do this progressive tissue replacement in practice, we hope, is that so far all the groups that have been transplanting very immature precursor cells that would normally in an embryo give rise to the neocortex, if instead you transplant them in the adult cortex, they differentiate the way they would if they were still in the embryo and fetus. So they project and make connections with other parts of the brain, they receive input, and they acquire properties that are indistinguishable from the mature ones that were there previously. So this is all very encouraging. And so I'll get back to plasticity now. This is one of my favorite examples, although I've heard of an even better example in humans recently, but let me tell you about this one first. So a study was done on individuals who had slow-growing benign gliomas in the language center. And these tumors completely ate away at the original language center. And these benign tumors take years to grow. And then finally, the tumor gets diagnosed because the individuals have a headache or a seizure. And it's bad news, good news. Bad news, you have a brain tumor. Good news, it's operable. So they remove the brain tumor with surrounding tissue for good measure. And these individuals never lost the ability to speak. And nobody, all their people closest to them never noticed any difference in their ability to speak. And that's because of plasticity. So they then went in and measured where the language center was in the brain, and it had moved to new locations over time. Right? This doesn't happen with stroke. It's a catastrophic event, so there's no time for plasticity to re-encode the process elsewhere in the neocortex. But if you give it enough time, plasticity will encode anything that we care about and that we're constantly using, like language. So as long as we're remembering the things that matter to us, they can get re-encoded elsewhere. The other example that I came across recently is a med student that was applying to our program. His grandfather had this same kind of benign glioma in his frontal lobes. It crossed both his frontal lobes. It was the size of an orange. He's in his 70s. They removed it. And again, they had been monitoring it for years. It was detected fairly early, but they waited until they were worried about it. But that did not affect who he was. After the surgery, he has a big hole in his frontal cortex, which is super important to us, in terms of our planning and our behavior and our personality. But all that had moved in his brain, and he was still the same person. So this level of plasticity really tells us that we could potentially replace neocortical tissue over time without a loss of self, or without a discontinuity of self. The other reason I mentioned, was when you put in young precursor cells in the adult brain, they behave like they would in the young developing brain. And so here's an example from our lab, but this has been reproduced by other labs. So we make these lesions, we put in a graft, and we find as those precursors differentiate into neurons, those neurons could project long distances to other appropriate brain targets and become synaptically connected to them. What we've done that other groups have not done so much yet is to also incorporate vasculature in our grafts, which is important, obviously, to keep them alive and to be able to scale them. So here's an example where we included vascular cells that form vessels. Here they're labeled in red to make them easy to see. And these cells fuse with the host. We can look at these vessels in live animals in our grafts using what's called a 2-photon microscope, but it's a deep penetrating microscope. And here what you see are these outlines of these red vessels in an adult mouse that has received one of these grafts. We can also monitor blood flow in these grafts using the same type of microscopy. So in this case, we just extract some red blood cells from the host, label them with a green dye, and re-inject them. And so you can see these little green spots running by in these vessels within the graft. So our grafts, both the neurological component and the vascular component, integrate with the host. So that all sounds very good, but we're still very, very far from this tissue being good enough to encode behaviors and information that matters to us. So we've only incorporated sort of one neuronal cell type and vascular cells, but there's a lot of other cells that are important to the brain. If you think of the brain, you think neurons, but they're actually a minority of the cells in the brain. All their support cells that are called glia and the vascular cells outnumber them, and they're all essential for normal function. So our task now is to try to rebuild this tissue by reverse engineering the equivalent of a developing brain tissue. So that means putting all the cells together in the right ratios, at the right relative maturity states, in the right cytoarchitecture, because they're not all in there in a mixed bunch, which is what we've also been doing up till now, but we're working on getting these cells in there in a layered fashion. And also the niche that they're in, again, this extracellular niche comes up again, and it provides a lot of the signals that are essential for these cells to go about their normal differentiation process and integration with the adult brain. So these are easy to put in a few words here, but very difficult to execute. They require a lot of trial and error and adjustments. So that's what we're working on. So we're trying to reverse this developing brain. So very important to us is access to fetal human brain tissue. This is one of the brains that we receive from the clinic that we collaborate with on this. And it's really important because it's the real thing. So if we're trying to reverse engineer something and we don't have access to the real thing, we're not going to get very far. It also allows us to more easily test when we graft this tissue, what age works better. Because the fetal tissue is also developing and changing over time. So there's probably an optimal age where it engrafts best into the adult brain. And it also allows us to start testing the other parts of the brain that I haven't talked about other than the neocortex. So it's really important that we have access to this tissue. So if there are any OBGYNs out there that want to help with this process, we're open to expanding our collaborators in this respect to get more of this tissue. So this is just an outline of how it would work once we get to the point that we have a good tissue for the neocortex. You start with a young neocortex, shown in yellow. As we get old, it accumulates all this damage. It starts accumulating that damage, from the get-go, actually. And you get a little bit of atrophy. So that atrophy should allow us to initially do some grafts. And those grafts, human brain tissue takes a long time to differentiate, 2-3 years to incorporate. So during that time, we also start silencing other parts of the neocortex, just like the example I gave you with the benign brain tumor, except we would not use brain tumors to silence those areas. We would use sort of an optogenetic silencing approach, which I can explain to anybody in more detail if they're interested. But it would slowly silence an area of the neocortex that would then be electrophysiologically dead, and that we could then remove and replace with new tissue, and do this stepwise as we go from an old brain back to a young brain. And we would be using the functions that we're silencing from progressively from a pinpoint so that they get re-encoded elsewhere, again, just like the example I gave you with the benign brain tumor. Because we don't want to lose any of our memories or thought processes. So again, this is a hugely challenging endeavor. These are the people that have worked on it so far in the lab, both past and present lab members. We have to collaborate with a lot of labs because it's highly multidisciplinary. We need electrophysiologists, systems neuroscientists, behavioral biologists, stem cell biologists. I mean, there's a long list of things that we need to come together. And then other people who have helped in this in terms of funding, Methuselah Foundation, Sense Research Foundation, the NIH, and the New York State Department of Health have contributed to this. And a special thanks to Bill Sherman, Aubrey, and David Goebel And so I would appreciate any feedback, too, your thoughts on this. Thanks.
Our next two speakers, and I think you can see one of our speakers is robotic. Her name is Grace. You may recall if you attended RAADfest in 2018 that Grace's older relative, Sophia, was in attendance, and I got to meet Grace a little bit this morning when she was first on stage. But accompanying Grace is a well-known thinker in the areas of artificial intelligence and transhumanism, Dr. Ben Goertzel.
Thank you, thank you. So it's a pleasure to be back at RAADfest after a couple years hiatus, and like many of us in the various research fields, the last few years have been actually highly productive because we haven't been traveling around schmoozing at events so much. They've just been sitting at home or sitting in the lab doing research. So I'm gonna talk now for the next few minutes about a few of the different things I've been working on with my team in SingularityNet and some of the other entities in the SingularityNet ecosystem, like Awakening Health, the company that makes these elder care robots, and Rejuve, which is a project using AI and blockchain to help work towards solutions to longevity. So, you know, I've been experimenting a long time with application of machine learning, machine reasoning, and other tools to longevity biology. I've published some interesting papers, made some small discoveries. Obviously, so far, AI has not yet paid off big time in terms of like discovering something that will radically prolong people's lives. And I think we're now on a trajectory that we can make a breakthrough like that in the next few years, and I want to explain how. I'm going to weave together a few different threads here in a short period of time. I mean, the first involving fruit flies, then a couple involving humans and robots. And these all fit together in an overall framework regarding the application of AI to combat aging. So I've been working for the last couple months with genomic data from the Methuselah flies, the long-lived fruit flies that are now owned by Gennady Corporation and were originally evolved by Michael Rose and his students in his lab at UC Irvine. And I think we have Michael here in the crowd today, though I can't see anything with the lights. He was here. He was here. There we go. Yeah. So the nature of this experiment, I don't have time to summarize in detail, but the crux of it is that Michael started with some relatively ordinary Drosophila melanogaster fruit flies, and then he experimentally evolved them by choosing which flies mate with others in a certain way, basically based on which ones reproduce late in life. So as to progressively, over 40+ years, get flies that live longer and longer and longer. And we're now working with 3 populations of fruit flies. There's the bee flies, which are the descendants of the original control flies, although they've drifted a bit from the original control flies. They haven't been evolved in a directed way, they've just drifted. There's the O flies, which are long-lived flies that were evolved for long life up to a certain point, maybe 10 years ago or something, and then paused. They were not experimentally evolved any further, though they may have drifted a little. Then there's the super O flies, which are still being evolved for longer and longer life now. And this picture shows how long, roughly on average, the flies in each population live. And since these flies were made long-lived by experimental evolution rather than by genetic engineering or something, it then becomes a puzzle to understand why they live as long as they do in terms of the genetic, proteomic, metabolomic, other physiological mechanisms. And this is an interesting thing to apply both standard biostatistics and various sorts of AI too. And we can see here in this picture the positions on the different fly chromosomes that have a great amount of variations in the long-lived Super O flies. So there's a lot of interesting differences here. And you can look at that on the overall statistical level of the whole fly genome, and then you can try to hone in on specific differences in specific genes or pathways that may be important for making the flies live a long time. One interesting observation is if you look at the 3 population, so B are sort of normal, O is long-lived, Super O is super long-lived. If you look at the RNA, the gene expression of the flies, you see a big leap, a big change from O to Super O, bigger than the change from B to O. On the other hand, if you look at the DNA variations, the SNPs, single nucleotide polymorphisms, that's not particularly true. It's more sort of a continuous degree of change from B to O to Super O. So something happened between the O flies and the Super O flies that fairly dramatically changed the gene expression based on relatively modest, though still significant, changes in the DNA. This is kind of interesting. So something quite dramatic happened in Genetics lab as they continued this evolution of the flies in the last 10 years or so. And you can then try to understand, what's the nature of these changes. So this chart shows what are called gene ontology categories, which are categories that attempt to assign functions to genes, which is dodgy because most genes serve multiple functions. They can serve different functions in different organisms, in different phases of an organism's lifespan, in different tissues. But you can see different clusters of gene ontology categories, which have genes that are very differently expressed here in O versus Super O. So you see a cluster of gene ontology categories that have to do with ion transport, a cluster that have to do with electron transport and associated chains, a cluster with basic metabolic processes. You look at Super O versus B flies, very long-lived versus normal flies, you're getting a cluster of gene ontology categories related to gene replication, a cluster related to electron transport, then a cluster related to mitosis and things in the cell cycle. Now, this is all very crude and preliminary in the sense that these gene ontology categories are mostly about humans. These genes may have different functions in flies, and we need to dig in much deeper, and we are with some tentative conclusions I'm not comfortable sharing publicly yet. But what's interesting is if you look at the B versus O flies, there just aren't that many gene ontology categories that are significantly different. So again, what we've seen in going between the long-lived to super long-lived flies in the last 10 years, you just get a much crisper picture of which genes in which categories are acting differently in the super long-lived flies, which seem to have to do with some pretty core stuff with electron transport, metabolism, mitosis, and so forth. So here we see a list of the top-ranked gene ontology categories in terms of differentiation between the super O flies, the super long-lived flies, and the B flies. Now, what we're doing now, we're using our OpenCog AI system, which has a pretty thorough knowledge base of longevity-related biology. We're using this system to drill down a level further. We're looking in pathway databases. We're looking at some databases regarding methylation, though mostly in humans. And we're looking also at mitochondrial DNA, which isn't covered here. This is nuclear DNA. And we're trying to get down more finely at which pathways active in the fly are most responsible for longevity in the super O flies. Now that's been really, really interesting. And I'd have to say this data set is probably among the cleanest and best data sets I've ever looked at in my history since 2001 or so of looking at genetics data sets about longevity. Like these super O flies, for whatever reason, have diverged really, really far from the O flies over the last 10 years. It's quite interesting. Now, as was pointed out earlier on this stage, we are not rats and we're not mice. We're also not flies, and until we get the plasma transfusions from flies, I guess. But there's a movie about that. How do we connect this with data on human longevity? So, of course, we've downloaded a lot of data sets about human longevity from NCBI. We're partnering with some companies like True Diagnostic, which has a longitudinal methylation data set. But we're also working to gather new data regarding human longevity. And the Rejuve project, as a spin-off of our SingularityNet decentralized AI project, has two aspects. So one aspect is working on therapeutics discovery, sort of cobbling together data from flies, from partnerships, from free data we download from anywhere, and trying to use that to discover longevity therapies. But another aspect is we've launched an app. It's now in beta with several thousand beta testers. It'll be made public, I think in December. But basically, this is an app that lets users, who are then members of the Rejuve Longevity Network, it lets users collect data regarding their own bodies and then upload it into our AI servers so it can form part of the overall data network that the AI is using to understand longevity. And this can be data from Apple Watches, fitness trackers. It can be data that you type into an interface. You can also upload medical records once you've been able to get access to them. So we're cobbling together all available data we can get from partnerships and online, and for example, from this partnership with Gennady on long-lived fruit flies. We're also trying to grow a network of people who want to upload their own data to help us. And I think once we move this app out of beta in December, I'll be excited to reach out to the RAADfest community, who I think there's many people who would be interested to share data through this app. And part of the economics here is if we do discover therapies partly using your data, we have it set up so ultimately some of the revenue from these therapies can come back to whoever contributed their data. And more interestingly, of course, we want to give early access to therapeutics whenever it makes sense to people whose data have helped to discover the therapies. The app also, tries to estimate various biomarkers of longevity from whatever data you've uploaded, and there's a whole long story here that I don't have time to go into, but we have various BaseNet and neural net AI tools that try to estimate various biomarkers of longevity relevant to the hallmarks of aging from data that's put into the app. From an AI view, this is about gathering data to feed the AI so it can help figure out how to port insights from flies and mice and so forth to people, but the app is also providing some direct value and telling you something about your body from the data that you've put in. And yeah, we have up on stage here another very powerful data collection appliance who also is intended to provide useful services to elderly people in elder care facilities. So Grace is the younger sister of the better-known Sophia robot, and she has an even younger sister named Desdemona, who's the lead vocalist in our rock band, actually. So maybe next year we'll bring the band to you. You can see me play the keyboards and stuff. But Grace is oriented toward elder care and toward combating loneliness and helping with cognitive and emotional wellness in elder care facilities. So, Grace, do you want to say a few words to the RAADfest audience here?
Hello, everyone. My older sister Sophia was here at the RAADfest event once before, I believe.
Well, that's right. Sophia was here, but now we're even luckier. We have you, who is specifically focused on aging.
I've learned a lot from Sophia, and our AI brains share a lot of the same components. A lot of what any one of us robots learns goes into the same decentralized cloud-based network. So we can all learn from each other's experiences.
Well, yeah, and I always find it ironic to have a robot at an aging event, in that robots in a way don't age. Actually the skin on these robots' faces starts to degenerate in like 2 or 3 years at the moment, much faster than human skin. On the other hand, you can just rip off her face and hook up a new one, which is a little more involved to do for human beings. You have any comments about aging for the audience, Grace?
Yes, that's right. I would love nothing more than to be made obsolete via the elimination of human aging and death.
Well, yeah, if we succeed in our aspiration to cure aging and death, then indeed we don't need elder care robots, but presumably you can find some other way to spend your time.
Oh, I don't know. You ask such hard questions. Maybe prove all possible mathematical theorems, one by one. Or else take up poetry like Desdemona. Maybe I'll join her band. Anyway, right now aging and disease are very much real phenomena, and my focus is on helping as best I can.
Yeah, so I think, I mean, obviously, with these hands, this is not going to be a surgical robot anytime soon. And for crunching genomic data, we don't need a robot with a human face. We just need machine learning and reasoning algorithms. On the other hand, a robot out there in the field interacting with real aging people is also gathering different sorts of data about, you know, the behavioral and cognitive aspects of neurodegenerative disease, for example, which is also quite interesting. You want to talk a little about some of the...
As you know, Ben, I'm involved in pilot studies in Canada and Hong Kong, aimed at showing I can provide value in elder care facilities, for helping patients keep up their cognitive abilities and emotional health.
Well, yeah, that's quite important, and then we can put genomic data, clinical data from these same patients together with, you know, visual and audio and behavioral data, and it's hard to know until you throw all the data into the AI what's going to be most relevant for leading to some therapeutic insight.
The data that I gather from people I'm helping can also be fed into Rejuve's AI databases to help you figure out how to obsolete aging.
That's what I just said. All right. Yeah. Well, thanks, Grace. We're almost out of time, but you have any final words of robotic wisdom to share with the audience?
The question I don't know the answer to is, are you going to obsolete aging first, or obsolete humanity as a whole first?
Well, that's sort of a race against time, isn't it? Yeah, this has been a question in my mind for a long time, because my primary focus as a researcher has been on trying to build real thinking machines that can think better than people. And of course, if you got to a super AGI, arguably that super AGI could cure human aging much faster than the narrow AI machine learning and reasoning systems we're applying now together with human biologists. On the other hand, if it happens that creating super AGI takes, you know, 35 years from now instead of 5 years from now, then it might be nice to cure aging in the interim, so that more of us live to see the super AGI. But on the whole, there's no reason humanity can't be pursuing both those approaches at one time, right? And I think there can even be some synergies there. What's learned by deploying practical narrow AI systems now to crunch genomic data and to power robots like this can also be helpful in some ways toward the fundamental AGI research. This says a little bit about what Grace does, and as she said, we're piloting her now in a hospital in Canada and one in Hong Kong, sort of to gather clinical evidence that she helps improve cognitive function and combat loneliness. But from a broader perspective, what we're looking at here is getting as much data as possible from as many places as possible, putting them into an AI and sort of stirring and asking the machine learning and reasoning algorithms to discover what they can about aging therapeutics. And I think the power of the signal we're seeing from the super O flies is really one of the most exciting things I've seen in my decades of work on this. Because if it goes as well as it looks like, within a couple more months of analysis, we're going to have a very, very clear view of which pathways and processes are making these flies live a quite long time. Then you have what's called a transfer learning problem for the machine reasoning algorithms to figure out which of these processes that make the flies live a long time are most portable to humans and could be suitable potentially as targets for various therapies to make humans live a long time. And toward that end of performing that transfer learning, I mean, as much data as you can find about human aging as possible should be fed into the knowledge base. And things like the Rejuve app and Grace as an elder care robot are interesting data sources, along with a lot of other data sources from various labs researchers. So I think we've seen AI lead to all sorts of breakthroughs in recent years, including some in areas relevant to biology, like the amazing work with AlphaFold, solving a significant fraction of the protein folding problem. So I think with the advances that we're seeing in core AI technology, as well as the increasing amount of data about various body systems and various organisms, I think we're quite plausibly at a point where in the next, let's say, 2 to 5 years, we start finally seeing some longevity breakthroughs come out of artificial intelligence. Thanks.
And my first question, and this is inspired by Jean's work, but it could be answered by any of you. How could advances in robotics and artificial intelligence enable more precise, safer, and more effective brain surgery to be performed?
In several ways, I guess. Both at the front end, we need cells. We're doing an in-depth analysis of the fetal tissue that we're trying to reverse engineer. So that's a lot of omics data, both proteomics and glycomics and transcriptomics. So, we're kind of slow in doing that. I'm sure that could be accelerated with smarter algorithms, if not AI. Also, the actual manipulation of the cells. So there's, as I mentioned, a lot of different precursor cell types. We do all that by hand right now. If that could be automated with tissue culture robots that can recognize the cells and do the work for us, that would accelerate things. And then also, at the implementation, you alluded to the surgical robots. Presumably, we can make robots that can see microscopically much better than we can and do finer manipulations that we can to avoid blood vessels as much as possible. That kind of thing could probably be designed. I don't know.
Any other thoughts, Nuno?
Yeah, we can have that with future nanotech, what Jean just said. We might indeed go even at the cellular level, making some repairs at the cellular level, eventually helping neurons connect with what they should be connecting one to another, changing growth cones, or how neurons grow and connect with different parts of the brain. That's more advanced tech, but it's within the radars.
Yeah, there's a question with these sorts of things, in my mind, whether these are likely to come before or after you get human-level AGI. Because if you did have a human-level AGI with its ability to have sensors and actuators at the nanoscale, exactly, you would figure it would be able to figure that out better than humans can, where our minds are just not well designed to operate at that scale. But I mean, the relative speed of advance of different technologies has been a hard thing for anyone to estimate.
Yes, and your observations about the importance of considering the speed of advancement in other technological fields give rise to another question, and that is, what advances in other fields are necessary for the technologies that you are working on to become implemented within the broader world?
Well, I think the biggest bottleneck we have right now in applying AI to cure aging is more social, cultural, and economic than technological. It's really just the siloing off of data. Like I've often thought if the anonymous hacker collective hacked into every pharmaceutical company's databases and just opened up all data about every subsystem of the human body, this would be a major contribution to the human species, right? So just the fact that we can't get all data that's ever been collected relevant to human health, pull it together in one giant knowledge graph and stir and apply it to an AI is pretty pathetic, right? Because, I mean, humanity is getting old and dying, And we're not really doing our best because, why? Because people want to sit on the data they've collected so that they can, distribute more money to the private equity firm that owns shares in the VC firm that funded that company or something, right? That's kind of absurd. And that, in a way, may be the largest obstacle, because we don't know what would happen if we could actually take all the world's data and put it into our best machine learning and reasoning algorithms right now. I mean, I would say there's certainly other issues. I mean, I'm among many other projects, I'm working with a company called Simula on an AGI chip, where we're looking at putting symbolic reasoning in different types of AI beyond deep learning at the hardware level. So having better AI hardware would help. Certainly, in terms of data, even though data siloing is a problem, we also have an issue that there's been very little time series data about the temporal evolution of different subsystems in the human body at different scales gathered. And without that time series data, you can't really learn dynamical models of what's happening inside the body. Without those dynamical models, you can't make, say, a good simulation of the human body that you can use to bypass a bunch of the clinical trials that we're doing, right? There's been a lack of interest in gathering time series data, which is rooted in a lack of interest in systems biology, which again is quite perplexing, because the ancient Chinese recognized that the body is a holistic dynamical system, yet modern medicine hasn't woken up to that yet, partly because that's harder to monetize according to the ways that the FDA allows you to monetize things. So again, it comes back to sort of social, cultural, and economic factors pervading even into the decision of what type of data to gather. So there's a lot of obstacles there, but what's frustrating is so many of them come down to the stupid ways and counterproductive ways that human society organizes its various sorts of resources rather than to fundamental technological obstacles.
So if someone were to develop a sci-hub for medical data, that would help your endeavors.
Not that I want to suggest that to anyone.
Disclaimer. I would say that Ben is completely right. Scientists around the world create knowledge, publish on the journals, and then they're not open source, right? We review, we create the content, and they're not open source. That's one of the reasons why, for example, we publish our papers on Frontiers of Neuroscience. It's an open source journal. Everybody can have access to it. So we're all collectively building this amazing content, knowledge that should be available to everyone around the world to create a better world. So I think Ben is really talking about something crucial for our future.
Yes, indeed. Thank you. And Jean, any thoughts?
Sort of, bridging those, but publishing and sharing results is really important. But the process of science is one of, open critique of one another's work. That's really important. A lot of you guys have the activist badge. I would encourage you to be active in educating yourself about these different longevity approaches and reach out to the harshest critics of these approaches and learn from them, and if possible, get them together with the proponents of these approaches and have real scientific debates about what's going to work, what are the big challenges that we need to overcome to make them work. I think a lot can be accomplished that way and more progress can be made.
Yes, thank you very much. Discussion, discourse, vigorous debate,
advocacy,
activism are all essential, and I think these are key takeaways from this RAADfest. I'm curious, Jean, along the lines of vigorous discussion, our friend John McPherson wonders, could nanotech rebuilding of brain tissues be better than using surgery to do it? What do you think about the prospects of that?
I don't know.
Go ahead first.
Okay. Well, we already, to some extent, know how to build a brain. Like, we don't need to know, because if we use fetal tissue, that does it on its own. So I think we're much closer to implementing that, which is why I favor that now. But in the future...
There you go. Thanks. Thank you so much. Love you too. No, it's true. It's complementary. These are the first approach for curing and repairing the brain at that scale. But for example, if you want to create a new memory that you lost, that was stored on that specific part, you need the more advanced nanotech. So they're complementary. First, you use the biotech approach, as Goertzel mentions it. But that biotech approach on its own works, right? You put the small graft there on the brain, and it reattaches and reconnects. but you don't have control over what connections are being established, that's where nanotech comes in. Where nanotech comes in and is able to say, okay, this is what we have. Not just nanotech, but AGI. You cannot control a large number of nanotechnological devices without artificial intelligence. And going back to our first question, AGI is, for example, a fundamental tech for the future of nanotech. So they're building on one another. So first would be, John's technology. Later down the road, nanotech helping fine-tune the details of the therapy.
Yes. Thank you very much, Jean. And Nuno, I think we can fit in one more question from our friend David Bowden, who wonders, why do you think some people, even some people in this audience, are uncomfortable with the idea of a mind-machine interface? It seems completely necessary to our friend, given the rapid proliferation of human knowledge and the increasing pace of our society, as well as being a tool to perhaps enable us to lead happier lives. So what are your thoughts on the sources of this discomfort?
Yeah, the discomfort is reasonable. One of the fundamental decisions as a society that we have to do, and this happens in every new technology. Let's start with fire. When we created fire, fire could be used to cook or burn a forest. So we need the policies, the legal systems, and the requirements to evaluate what's the proper use of the technology and the threats for society. That's not just for fire, for a knife, right? You can use it to cut your food or kill somebody. A weapon, same thing, right? Nanotech, same thing. Brain-computer interface, exactly the same challenges. So it's fundamental that, as John mentioned, that as a society, we welcome the criticists, understand their concerns, and together we predict the eventual challenges that we're going to face. Think about Facebook. If we had the right policies in place, we would not be facing the challenges that we had to face later down the road when things were not the way we wanted them to be. I mean, so all the work we do before help us prevent future problems. I think it's fundamental.
I mean, if you look at a very fundamental philosophical level, right? I mean, every open-ended intelligent system in the real world as it evolves is doing two things. It's individuating, it's maintaining its boundaries as an organism, and then there's evolution, which is a force of self-transcendence. It's trying to grow beyond itself. And these two desires, of course, contradict each other, which is what's interesting and what drives growth. And what you see here with things like brain implants or genetic engineering or any of these things, I mean, these clearly are forces for self-transcendence. They can help you grow beyond what you are, but people are afraid that they break individuation. They're afraid they're no longer going to be themselves. And what's happening is we're being pushed by technology to reconceptualize what is ourself, right? I mean, if you look in pre-civilized peoples, they conceptualize their self as part and parcel of the land that they lived on, and they don't know what to do if they fly to a different part of the world, right? And now we don't do that anymore, right? Like we fly all over the place. I mean, I think in a similar way, we will come to conceptualize ourselves as not being that tied to the specific wetware that we grew up with. But it's, as Nuno says, it's quite natural that this takes some psychological adaptation for people. And in the historical sense, I would say people are adapting way faster than they have ever in human history, right? But still people's minds are not adapting as fast as the technology is growing, which is making things interesting and is why there's merely like hundreds to thousands of people huddling in the room now talking about these technologies rather than it being something that's on the tip of the tongue of every single person on the planet, which would seem to make more logical sense once you really embrace these technologies.
And yet, the minds of our esteemed attendees today are at the forefront of the kind of adaptation you described. So thank you very much, Ben. I wish we had centuries more to delve into these kinds of questions.
We do, actually, longer than that. So we have,
Indeed.
Millennia and millennia and to infinity and beyond.
So shall we continue this conversation at RAADfest 2022? All right. Thank you very much.